This is our presentation on the comparison of environmental interactions between Hylobates lar and Sagrinus mitis. We wanted to know whether Hylobates lar switched between trees more often than Sagrinus mitis. Before creating our hypothesis, we did some background research to find out more about each species. Hylobates lar, more commonly known as the white-handed gibbon, is known as a true brachiator. This means that it has a long arm-to-leg ratio and spends most of its resting and socializing time in trees. Hylobates lar is a frugivore, with up to 50% of its diet consisting of fruit. Sagrinus mitis is known as the red-handed tamarin and is a primate that lives primarily in northern Brazil, Guyana, French Guiana, and Suriname. Because their diet consists of mostly insects, fruit, and plant exudates such as sap and resin, Sagrinus mitis can spend up to 60% of their time foraging for food around trees.
Based on our research, we hypothesized that Hylobates lar seems to depend more upon trees for shelter, nourishment, and socializing than Sagrinus mitis. Hylobates lar will spend more time moving through trees than Sagrinus mitis in a set period of time. In order to test our hypothesis, we decided to observe each species independently for 2 trials of 20 minutes. During this time, we would note the amount of trees switched between members of a species and calculate the average. We observed members of both species in the Santa Ana Zoo. Here, you can see the layout of the Hylobates lar habitat. The habitat for a Hylobates lar is split into 3 different heights. We counted different branches of rope as trees as well as the enclosure contained both ropes and trees. The swinging spots were distributed evenly throughout the entire gibbon enclosure. There was only 1 gibbon. The Sagrinus mitis enclosure was slightly different than that of the Hylobates lar. As you can see, the enclosure was made up of multiple trees with many interconnected branches. However, 3 basic levels of height were still noted to be present in the red-handed tamarin enclosure. There was a family of 5 in the Sagrinus mitis enclosure. Our ethogram measured once every minute for 20 minutes over 2 trials. When averaging the results, we eventually reached an average of 144 branches swung per minute with the Hylobates lar, an average of 17.5 branches switched for each member of the Sagrinus mitis. As you can see from our pie chart, Hylobates lar swings between far more trees than Sagrinus mitis as per our hypothesis. We believe that Hylobates lar switched between many more trees than Sagrinus mitis because of their different lifestyles. Although both rely on trees for nourishment, Hylobates lar's lifestyle mandates that it stays in trees for the purposes of social interaction, rest, and travel as well. Thus, it would make more sense that Hylobates lar switch between more trees. An experimental error that may have affected the quality of our measurements is the method in which we took our sample data. The cage of the red-handed tamarins contained 5 tamarins and so required measuring 5 different movements at once. This may have caused the data to be missing some points. The experimental data may also have been affected due to small sample size and limited trials. Only 2 trials were conducted, and with only 1 Hylobates lar and 5 Sagrinus mitis. The small sample size may have biased our results. If this experiment is to be revisited again in the future, more trials with more accurate recordings would be used. In conclusion, Hylobates lar spends more time swinging through trees than Sagrinus mitis does. To conclude, here are the sources that we used to research data on both the species of Hylobates lar and Sagrinus mitis.